We should get going, huh? Okay. Hey guys, I'm loud, so I have to get away from the microphone because, no, I'm okay? Okay. Okay, good afternoon, everyone. Welcome to CBET's Symposium this afternoon. It's called the Stack Family Center for Biopharmaceutical Education and Training at the Albany College of Pharmacy and Health Sciences. I just want to give you a brief overview in a few minutes of what we're doing at the center. We're really a multifaceted center. We're established to do a lot of education and training programs, and we're also there to establish a lot of collaborations with industry. And so if you look at on the slide on the board, you can see that we have emphasis on workforce training. And today's panel discussion will be on workforce, the importance of workforce, and how we can prepare tomorrow's workforce today with all these new technologies coming about. And you're going to hear some of them today from our speakers. There are exciting new technologies that we never heard about. And for that, you need workforce, and that is something that we have to tackle because these technologies are going to be into action to make different types of products. So one arm of the center is building workforce training program, and the workforce training program is really in a two-way. We can advertise programs. You go on our website, you see we have a number of well-defined, well-designed, with good instructors training programs that's open. We're calling it open enrollment programs. You can register from anywhere in the world, especially with online parts of it, you can register. But the on-site program is the hands-on activities that will be done at the center. We also have a one-year master's degree program with collaboration with our academic department heads and our faculty. It's a really fantastic master's degree program for our students and for the fact for anyone in the professional life who may want to get into this degree program to get a master's degree and start a new career, possibly in the biomanufacturing industry. With those two, we are also starting to look into doing some projects at the center. Contract research, we call it. And please remember, this is not CRO, this is not CDMO, this is just basic contract research in a non-GMP environment for any industry or academic institution who may want to test out a project, and we have the equipment and the facility and the staff to do it. We can do it for a fee. So that's basically our activities explained in that slide, so to see it. And we have a couple of slides. Some of you have been to the opening yesterday, and I saw the labs. For those of you who haven't been there, please come by anytime you want. Just let us know a day or two in advance so we can get you all the permissions that you need to have to visit the facility. Today's program is divided, like I mentioned, into two sections. Section one is presentations by some of our really high caliber speakers that I want to thank them for coming to participate in this program and join us in our celebration of opening this beautiful center. And that is supposed to take about an hour and a half from 2 minutes from now. I got still 2 minutes, Julian. Julian is going to catch me, with my time, And also what we're going to do, we're going to have 10, 15-minute break. So my suggestion is that in the crowd, if you have a question for the speakers, we're going to have time for one question or maybe two questions at the most. But we have online people that are joining the program, and they may want to ask a question. So I say, since we have a 10, 15-minute break, and all our speakers will be here because they're going to come back and join us for the panel discussion, ask the question during the break time, one-on-one. It will be easier to allow online people to ask questions. If there wasn't online, then that's okay. We can go ahead with the question. After the break, we come back, and we have formed a really great panel from industry, both types of industry, the suppliers industry and the biomanufacturing production industry, and we have academia. We have really great names in this panel, and we're going to discuss where we are with the workforce development, how we're going to get the workforce that's needed for these growing companies and this growing industry. And it's going to be open. We're going to go through that when we come to that part of it. So I'm not going to spend time on that. When we get to that part, we're going to talk about how the setup is and how we're going to start asking questions and stuff like that. So with that, I'm going to turn it over to Dr. Julian Rosenberg. Dr. Rosenberg is our associate director. He's also the director of the PSM program, the master's degree program. And he is chairing the session on the presentation. Dr. Rosenberg.
Thank you, Dr. Rashid. We're very excited that CBET can contribute to the growing ecosystem in New York City, or New York State and the city included, that we have in life sciences and biopharmaceutical industry. You'll see from the speakers that we have today, there's a great representation and really show that New York is cultivating a very fertile ground for biomanufacturing innovation. And this past year, we've really changed the way we've all thought about vaccines and biotherapeutics and their impact on global public health. So with that, the industry has made some great strides, and I think it's set the tone for the research directions going forward. With that, we're thrilled to have three leading perspectives in this area to talk to us both from industry and academic perspectives on recent developments and outlooks for the area. So first we have Dr. Sally Temple, who will be joining us virtually, and she'll share some of the incredible work that she's doing with stem cell therapeutics at the Neural Stem Cell Institute. Next, we have Ronald Bates from Immunovant, who will be offering an overview of how protein biomanufacturing has evolved over the years and what technologies exist on the horizon. And lastly, we have Professor Matthew Delisa from Cornell, giving us an update on what his group has been doing in the area of glycobiology and cell-free biomanufacturing. So as Dr. Rashid mentioned, we'll have time for one or two questions. And we're really excited to have Dr. Hassan Medinia taking questions from online. So please do type those into the chat, and he'll be reading them out loud in person. If you are here with us in attendance, you can come to either of the two microphones set up. So with that, I would like to introduce our first speaker, Dr. Sally Temple. She's the scientific director and co-founder of the Neural Stem Cell Institute, where she leads a team of researchers to develop therapies for eye, brain, and spinal cord disorders using neural stem cells. Dr. Temple's group is building cell culture models to study neurological diseases that inform our understanding of disease mechanisms and help identify novel therapeutic strategies. Her work has been recognized with several awards, including the MacArthur Fellowship and the Ellison Investigator Award. So please help me in welcoming Dr. Sally Temple.
So thank you so much, and it's just such a pleasure to share this very exciting opening, and what an important job you're doing to begin to find the workforce that is needed for all of these novel approaches to diseases, to therapies, and I'm going to talk today about our work using stem cells and to advance therapies for the central nervous system, that is, for the brain, the spinal cord, and the retina. So I hope you can see my slides okay? Yes? I can't hear, but I'm hoping. I'm just going to go on, so. Okay. So I just want to point out, these are my disclosures before we get going. Okay. So I wanted to start by introducing our institute, the Neural Stem Cell Institute. We're a nonprofit research organization actually founded in 2007. And here we are, we're in Rensselaer, New York. NSCI was purpose-built to translate discoveries in stem cell research into new therapies for neurodegenerative diseases. And we have about 35 wonderful researchers dedicated to combating diseases of the central nervous system. So today, I'm going to talk about two different projects that we're doing that also represent some of the new strategies by which stem cells are advancing these new types of therapies. So in the first one, I'm going to talk about our work on modeling diseases that impact the central nervous system, and specifically tauopathies like Alzheimer's disease. And in the second, I'm going to talk about our work developing a new cell replacement therapy. So starting with that tauopathy modeling story, I just had to acknowledge that this is a highly collaborative project. It was funded primarily through the Tau Consortium and the Rainwater Charitable Foundation, also Cure PSP, the NIH, and AFTD. Without them, we couldn't have done it. We have a fabulous group of investigators around the country who help us, and these are the great scientists at NSCI who have helped with the project. Now, tauopathies are diseases in which an intracellular molecule that's inside of neurons, it's called tau, becomes abnormally aggregated. It becomes modified abnormally, highly phosphorylated, and then the neuron dies, and that causes a lot of death, especially in the cerebral cortex in the brain. So the part of the brain that's responsible for memory, learning, integrating thoughts and sensory input. So once this happens, of course, we all know family members or friends who have these devastating diseases. Now, it turns out that tauopathies are also, in some cases, genetic, and one of the causes can be mutations within the gene that encodes tau, and that's called the MAPT gene. And that can lead to an early onset disease, frontotemporal dementia. Now, patients with tauopathies typically have decades of normal life before clinical symptoms appear, and we really want to understand what is happening during that period before the clinical symptoms. We need this information to understand the disease process and to determine how to combat the disease. And we know that if we understand those early changes, then we can intervene early before there's been a lot of damage in the brain. So researchers in the lab need models to study. And one of the revolutions in stem cell research has been the introduction of what are called induced pluripotent stem cells, or iPSCs. To make these cells, we take a sample from a patient. They generate these induced pluripotent stem cells that can produce any of the cell types in the body. So it's really profoundly useful. In our case, we're interested, of course, in making brain cells and then using those to model disease and hopefully generate new therapies. So in order to really model disease, we want to have like little pieces of brain-like tissue. And the way we do that is to grow these stem cells, not in traditional two-dimensional cultures, but in these little round three-dimensional spheres, a couple of millimeters across. We call them organoids or brain organoids. So we like to say they're small but powerful. They are much smaller than the human brain, but they have the neurons and the glial cells, the cell types that are affected in tauopathy. So here's that affected region of the brain in the cortex. And this is a schematic of the different types of cells that we expect to see. And here we have sectioned the organoids and done staining with antibodies that demonstrate that by about 4 months, the major cell types that we know are affected in disease are in these organoid models. So organoids are valuable as model system because they contain the human brain cells, so they're closer to the actual patients. But we can also scale up. We can generate thousands of these for study. And we use them in two main ways: as a discovery tool, but also to test candidate therapeutics. And I'll show you some examples of both. So the most effective way to accelerate progress is to work together in collaborations. And this project benefited from both collaborative support from QPSP and the Tau Consortium, and also from wonderful researchers at different labs in the country who specialize in Tau biology. So what we did is centralize organoid production here at our facility. We made the organoids and then we shipped them out to each of those participating labs, and they did the experiments that they are best at, looking at different aspects of the biology. And what we're doing is comparing organoids that were made from patients and organoids that were controls, so that we can see what the mutation causes. That's the critical thing. So in the end, we made and shipped out over 6,000 of these little organoids. And I wish I had a picture of that. I don't. And I have a picture of 6,000 baseballs. So you see, it's a lot. So just to summarize some of the key findings from these studies, and I don't have time to go into all of them. It was actually published August the 19th in the journal Cell, if you want to check it out. But we discovered a lot of disease-associated changes in specific genes and pathways that occurred before we saw any cell death in the patient organoids. And so what we think now is that we have a library of changes, of new discoveries that have come from studying this model. And we're going to be able to test those out to see if some of them are critical, and then hopefully to be able to target new pathways for new therapy development. So earlier I mentioned that we were using the organoids for discovery, which I just explained to you, but we're also using them to test candidate therapeutics. Now, when we're developing a new drug, we want to know that it's safe and that it's efficacious. And normally we do a lot of animal studies and then go into patients. Now, in addition to those studies, we have these human 3D organoids that we can use. And I know from conversations with FDA scientists, they are also watching very carefully the advance of these new human stem cell models and how they can help in that drug development pathway. So I wanted to highlight the work of one of our investigators at NSCI, Dave Butler, and his fabulous team of researchers. And they have been developing a new gene therapy that depends on a type of antibody technology. So we call these Tau-lowering intrabodies. Now, I mentioned to you already that abnormal tau is seen inside of cells. And the toxic molecule should be ideally removed. So in order to do that, we have to think of ways of getting inside the cells. And the antibodies then that we're delivering, we deliver as a gene therapy. And the antibodies then are bifunctional. They can bind the tau on one side, but at the other side, they have a degron that binds to the lid of this organelle inside of cells, which is called the proteasome. Or you can think of the proteasome as the sort of incinerator in the cell. It's involved in breaking things down, getting rid of them, and we need to get rid of that toxic molecule. So the idea is bind tau, push it through the proteasome, lower the tau, and that then should lower the toxicity inside the cells. So just to show you that this approach works, I want to show you this system, this test system in which we have the toxic tau molecule, which is tagged with a green fluorescent protein, and we've expressed that inside of cells. And then for some of these, we also include the engineered tau-lowering intrabody. And we've called these V, F, and N PEST, because that's just as the names of the constructs. So if these work, then you should lower the amount of green, you should see less green. And I think you can clearly see that while a control is brightly green with that pathological tau, once the intrabody is introduced inside the cells, it's very effectively lowering the tau. And you can see that also in this Western blot. So we're very excited to now push this ahead. We're going to be initiating animal studies as well to really make sure that this is safe and effective. So I just talked to you about stem cell manufacture. I talked to you about gene therapy, and gosh, we think about all of the new advances now in gene therapy that are going on, and the need to develop the workforce in order to address that need. And now I'm going to talk about using these stem cell products as therapies themselves. This is a cell therapy we're developing. It's not for the brain, it's actually for the retina. And the reason that, that there's a huge unmet need here, we're addressing the disease called age-related macular degeneration. So this affects more than 1 million people in the US alone, with huge costs to patients and their families, as well as economic cost. 1 in 5 people have AMD. And as the population is aging, as you can see here, the risk of getting the disease is dramatically increased with age. So you can see that this is a huge burden. Patients with AMD have a neurodegenerative disease in the retina. So there's the eye, and then the back of the eye, the retina. These are the light responsive cells, the photoreceptors. And this little layer of cells at the back of the eye is called the retinal pigment epithelium. And these are the cells that become damaged in age-related macular degeneration. And when they die, the overlying retina dies, and that results in loss of vision. And so patients with AMD lose vision actually in the center. They have peripheral vision, but just imagine if everywhere you looked, there was a big black hole in the middle of your vision. It means that you couldn't read. You can't recognize the faces of your loved ones. You can't easily take your medication, can't watch TV, all the things that are important for normal life and for independence, these patients lose. Just think about what it would mean to you if you couldn't read, right? So how do we improve the vision for those patients? I just told you that The retinal pigment epithelium is the layer of cells that die. Is it possible to renew that? Several years ago, we discovered that in the back of the human eye, in this retinal pigment epithelial layer, there is a stem cell that is dormant. What we found is that we can take those cells out and put them in tissue culture, and you can see all those little pigmented cells here, and that after about 12 days, we see growing colonies of cells that go on to divide and virtually take over the culture, and then they differentiate into these beautiful, newly formed retinal pigment epithelial cells. So from a single donor, we can make 5 times 10 to the 8 cells. That's a lot of cells when you think that the central retina is only about 50,000 cells. If we could replace those in patients, this would be wonderful. So we made these cells and we decided to go ahead and do some animal testing. And the first challenge we found was, how do you test vision in a rat? Well, it turns out this was solved by Glenn Prisky and Naziel Lamb down at the Burke Research Institute. They invented this machine which has 4 computer monitors, has a platform inside. You put the rat on the platform, and then these computer-generated bars rotate around. And just like if you're watching tennis, if you see them, your head will move, and they can measure the head movement in the rat very accurately. They can make these balls very thin, and at one point the rat can no longer see them because they're too thin, and at that point the head doesn't move. So it gives you a measure of visual acuity. It's also called spatial frequency, but just think of this as good vision at the top and bad vision at the bottom. So here's an animal that we use for this, has a degeneration in the retina. And an unoperated animal over about 6 months or so of life will show dramatic loss of vision, shown here over this time course. If you put in just vehicle, what the cells are in, it doesn't change. But if you use the cells, now in this animal, you can preserve vision all the way out as long as we've looked. And it's very similar, this high level, to the high level seen in normal control animals. And of course, we did this in both females and males. So this is exciting. It shows that the cell that we're making can rescue vision in an animal model. So we went on with this discovery. We developed a GMP manufacturing process. We then created clinical grade master cell banks. And the idea now is that we're going to be injecting those subretinally into patients who have macular degeneration, targeting that central part of the retina that shows disease. So these are the steps we've gone through. We filed our IND and we were allowed to move ahead. So we plan to be starting this clinical trial at the University of Michigan in about 6 months from now. So we're very excited to be at this point, and couldn't have done it without New York State stem cell funding, NIH funding, and also our partner, Luxa Biotech. We're very happy about this. I just want to stop at this point and acknowledge all of the wonderful people who contributed to this project on the RPE transplantation. It's a huge amount of work. Not everyone is mentioned here, but I think you can see how going from that first discovery right out now to planning the clinical trial, there were so many people who, without whom we could not possibly have done it, or without the support that I mentioned as well. And, we're excited to finally see this project and humbled to see this project now be tested in patients. So I want to thank you for your attention, and I think we have a few minutes for questions. If you have any questions, I'm happy to answer.
Thank you, Dr. Temple. Really impressive work. We do have time for a question, so anyone in the in-person audience can walk to the microphone and please ask. Nobody? Okay.
So I have one question. Thank you very much, Dr. Temple, for the great presentation and, of course, the very promising and great work. Did any of the test animals get cancer from the injection? Is that still a concern, or that has been bypassed?
Very, very important question. We had to do a lot of testing for tumorigenicity. Now, in our case, this is an adult stem cell, and adult stem cells don't have the same risk as if you're starting with a pluripotent stem cell. Nevertheless, we did a lot of testing, and none of the animals, and there were over 300, had any signs of tumor formation.
Wonderful. Thank you.
Yeah, it's an important question.
Excellent. Well, let's thank Dr. Temple again.
Come and visit. If anyone wants to come visit us, we're happy to have visitors. All right. Take care. Bye-bye.
Okay. I would now like to introduce our second speaker. We have in person our good friend, Dr. Ronald Bates, who is Vice President of CMC Process Development and Manufacturing at Immunovant, where he leads projects to produce novel immunotherapies. Prior to Immunovant, he led the Manufacturing Science and Technology Group at Bristol Myers Squibb, and before BMS, he led the Process Development Group at Allergan. He started his career at Pfizer, purifying small molecule moieties, and Dr. Bates is very active in the biopharma industry, sitting on numerous conference and editorial boards. Today, he'll be providing an overview of protein manufacturing, including infrastructure improvements, new technologies, and a forward-thinking outlook on where the industry is going. So let's give a warm welcome for Dr. Bates.
Awesome. Well, thank you, everyone, for actually attending. Otherwise, it's just me talking to myself, and I do that enough. I don't need anyone else's help with that. And thank you. Camille is here somewhere, and Julian and company. Thank you for inviting me to come speak. So my talk will be slightly different than the last talk in that I'm going to veer away from heavy scientific theory and real therapeutic stuff. And I want to focus on how do you actually make it and where do you make it more so than how do you make it. As everyone knows, you can do a lot of work in a lab, but if you can't bring it to a patient, it's kind of useless. So what I'm going to try and do is help the professors like Professor Delisa there and the different institutes bring their molecules to patients. Okay, so title slide. And the way I'm going to do that, first I'm going to just make a brief introduction of things, and then I'll go through a couple of topics, and I'll do so in a fairly rapid fashion. Different protein manufacturing, the way they manufacture, and in general, 75% of the proteins out there are antibodies, so my talk's going to focus mostly on therapies that are, or processes that are using therapies that are mainly antibodies. However, do realize that any protein would fit into this, and for the most part, viruses will fit into this fairly nicely too. And there's really two types of facilities that I'll talk about, as you can read up there as well. And then at the very end, because we are at an academic institute, I did want to talk a little bit about the future, and I am going to just talk a little bit in something called Industry 4.0. In different places around the different industries, they're way ahead of us. And the reason pharma is behind in all this is something I've always wondered throughout my career. In the aerospace industry and the nuclear industry, you know, people talk about error margins and you know, success rates and everything, and there's so many nines after their 9.9, it's incredible. And we get to biotech and it's good enough. So I do want to talk a little bit about Industry 4.0, but realize our industry is still very, very much in its infancy. Okay. So I don't necessarily need to go through my background, so I'm not going to, with one small exception. This is about New York State. And I did do my undergraduate just upstream here at RPI as well. And I have lived most of my adult life in New York State. So I am one of those homebred, although I did grow up on the other side of the Berkshires in Massachusetts, but we don't need to talk about that. Anyways, so there's my background, as Julian mentioned. So what do I mean when I talk about biologics manufacturing or protein manufacturing? And essentially, you know, I'm luckily, well, people in the back can't throw anything at me anyways, but luckily there's no food allowed in this, so no one can heckle me here, because I don't think the techniques have changed in my career, and probably dating even further back than my career. Protein manufacturing is essentially the same thing. You know, you grow cells in stirred tanks, you use pressure or chemicals if you need to break the cells, you harvest either extracellular or intracellularly what's there, you clean it up using a very few techniques, centrifugation, filtration, you further purify it, removing those tight to, you know, closely related impurities using chromatography and maybe some filtration, but mainly chromatography. And then at the end, you pass it horizontally across or tangentially across a filter, you exchange out whatever buffer it's in, and you put it in your formulation. And that's pretty much it. So thank you all for coming. No, but realistically, that's what we've been doing in this industry for as long as people can remember. I'm not going to tell you anything different. So if you remember anything, there's your protein manufacturing, okay? However, each individual technique, there has been some monumental improvements. And, you know, we could talk all about it, and we only have 20 minutes. So, actually, sorry. Let me just verify that. So we only have 20 minutes to talk. So I'm not going to go into each and everything, but realize right down the road, controllers, process analytical techniques, how we manage, how we monitor stuff, filtration, chromatography, the way we run, batch versus continuous versus semi-continuous, disposables. You know, we use plastics now more than ever. Hopefully there's not too many environmentalists in the room. Our industry is looking at ways to recycle and reuse better. Right now it's not so good, so again, we won't want to talk about that. But we are looking to get rid of paper and go to electronics. So that's not quite offset, but it's something. And then more importantly, from an engineering perspective, and I am a chemical engineer, is getting away from manual and getting into automation. I would much rather be sitting at my terminal pushing a button rather than going out there and twisting a valve, right? That's what I want to see going forward. We heard a little bit about some of the newer therapies like cellular gene therapies in the previous talk from Dr. Temple. I'm not going to talk about that, but realize that's probably the future of manufacturing, going away from big tanks to small, personalized medicines. And that's absolutely fantastic and fascinating to me. Again, I don't have enough time to talk about that, maybe at a future symposium. So, as I mentioned, a long time in protein manufacturing. We look at a couple of them, Epogen and Rituxan. We're talking in the 80s. I know a lot of the students don't recognize the 80s, but realize there is a lot of work that was done in the 80s on this. And that really paved the way. I mentioned earlier the different types of processes. You can just see it up there: inoculum, fermentation, disruption, purification, formulation. Like I said, that's really the nuts and the paste. Everyone can hear me, right? Okay, good. Again, just making sure I'm not talking to myself more than normal. And I did also want to make it clear that if you look online, you'll see the data. You're probably looking at 75% of the over the past 30 years have been antibody or antibody-like proteins. So that is, if you're going to study one thing, if you want to get into the industry, if you're good at that, there's probably a place for you in the industry. And I mentioned what has changed over the years, so I don't want to go back into that in too much detail. Dogs, I realize now that I'm looking at that, it's probably not a familiar term. Diaphragms, O-rings, and gaskets. So those are the connectors, and those are the sealants that you use in big tanks or among tubes. Sorry about that. I try not to put acronyms in, and I just realized that one slipped in, so I do apologize. Again, just to kind of take a look at things. So these are a picture from a former company that I did get permission to use when I was with that former company, but nonetheless, the facility on the left is a brand new facility. I was walking through it, touring it. Those are 15,000 liter tanks. It's a stainless steel in origin. And the one on the right, those are 5,000 liter tanks. So again, these are fairly large stainless steel tanks. If you go across the industry, if you do a Google, most of your existing facilities, say 5 years ago or more, are all stainless steel based. And there's a lot of reasons for that. The most obvious one is it was just cheaper at the time to do that. And the technologies were better at the time to do that. And then the most important one, from my opinion, if I want to go big, I still have to go stainless steel. Right now in industry, we'll get to that in a second. So if you're looking to make a lot of protein, you have to build it in a stainless steel facility, okay? But we'll get to more of that. Also, contrary to what people may tell you, they're very cost effective. Stainless steel, you have a high outlet, right? You put it out right off the bat, it costs you a ton of money to get that thing up and running, and then it's very little maintenance after that. As everyone knows, stainless steel is really resistant to most chemicals. They last a really long time. They're easy to pacify or to sterilize, and really they're easy to manipulate. You can change a straight pipe and put in, ebbs and flows, whatever you want. Really works nice. So they're very common, and they're going to be common. So just understand, I'm not someone who's going to tell you go to disposable, throw away all your stainless steel. I think there's a place for a lot of things. And if you want to go big, go stainless. I think, my ballpark is if you're going to go more than like 10,000 liters, today you're still going to build a stainless steel facility. Okay, so just order of magnitude. Turn a little bit. The opposite of that is a disposable facility. And I'm using the words disposable really to mean intended for single use. So your plastics that are bags, bioreactors, tubes, things like that. I've been touring the industry for the last 10, 15 years looking for people to manufacture my molecules for different companies. And I've come across really thinking most people who make molecules for someone else, these contract manufacturing organizations, they really should be disposable. It's really to their best interest. And I see a lot of new construction in the contract manufacturing business, and almost all of it is disposables. So just kind of think about that. Also, it's usually small, 1K, 1,000 liters, 2,000 liters, 5,000 liters. And there's a lot of reasons for that. Most of it is the sweet spot for CMOs is clinical. Clinical is typically want to go fast, small campaigns, and do a lot of different molecules, right? Because if I have one customer, that's good. If I have 50 customers, that's even better. Because as you guys know, throughout the drug development process, not all ideas make it to a drug. So from a CMO point of view, they want to play the lottery like everyone else, and just keep going, right? So if we take a little bit of a look at single-use bioreactors, there's a couple of different ways of doing it. One is you just get a really big bioreactor and you process it. Other ways are you get smaller bioreactors and you do tandem runs, combine at the downstream, and then go one downstream. So that's a way you can look at scaling up without actually scaling up. The way I look at it, it's the chemist way of scaling up versus the chemical engineer's way. You guys all know that, right? Chemists, if you go to a chemistry lab and you ask them to scale up, they'll give you five more vials or 5 more beakers. If you go to an engineer, he'll give you 1 bigger beaker. So this is, I think, the chemist way of scaling up. And I do recognize I'm at a chemistry-focused school. I was hoping that would go over better. But anyways, nonetheless, so there is different ways of looking at it. I've actually just last week was at a facility that had both options. It had 2 2,000 liters and 1 5,000 liter. And they both went down to the same downstream processing. So it's kind of interesting to see the different options just from that one facility. The other thing to note, there are disposable chromatography columns. And in fact, since we're in New York State, the biggest manufacturer of them is right down the road, Repligen, sorry. And they make columns up to 80 centimeters in diameter. So you can think of an 80 centimeter column as a pretty big column, and it probably weighs close to 600 pounds when it's packed and ready to go. So disposable, they're not thrown away after one use. They're usually used multiple times, but these do exist. Some resins make it less advantageous because they cost a lot. Protein A resin for antibodies comes to mind when you're looking at $10,000 per liter. Probably don't want to use that once and toss it. So there are some good and some bad with that. Okay. I just wanted to kind of take a couple more pictures. These are not mine, these are industry. I'm sorry, these are Google stock photos that I took. But I did want to kind of point out, the one thing with stainless steel is you can't really see anything. So there's a huge disadvantage. Often you have a little tiny viewport at the top, and that's how you have to do everything. And the reason I bring that up is if you're doing cell culture or fermentation, you oxygenate or you aerate, and one of the things that happens is you form foam on the top of it. And in the stainless steel, you don't actually know when to add anti-foam. So there is some give and take and some reasons, additional reasons why disposables are better. Again, we won't get into that discussion today, but there are some goods and some bads. And I just wanted to kind of give you guys an idea of a manufacturing facility again. All right. What I think the future holds is hybrid facilities. I think there's a really good use of existing facilities and an opportunity to improve them greatly. And I've listed out a couple of ways here. I used to work at a facility in East Syracuse that was very, very old, and it was archaic. And every time we had a problem, it took a ton of time to fix until we started going to this approach, which was rather than replace all the stainless steel arms that had ebbs and, because as I mentioned, it's very easy to work with steel. It's also fairly permanent. So if I put a bend in a line, that bend's going to be there for a long time. And now I've just added a dead leg. Somewhere around there's an opportunity for microbes to grow and things. So one thing you can do is as you're improving your facility, take out the stainless steel, that's okay, the pipes, the tubings, the connectors, the things that are small and easy to replace, and go ahead and use disposables. There are some benefit in that, and I've seen some really powerful use of that. The other thing to do is all cell cultures roughly start the same. You take a vial out of your -70 freezer, you thaw it at room temperature in a water bath, whatever, and then you slowly start growing it up in bigger and better containers. For the most part, the beginning 10, 15 days, 20 days of that are all done in very small tanks or beakers. Why don't you just use disposable for that? Why do you have stainless steel? So those are the places that I think you can make a lot more efficiency if you were to look at running a hybrid facility. And I have seen this work pretty well in a previous life, but I'm really waiting to see it start taking over some of these really big facilities with 20, 25,000 liter reactors. Because if I can give you a 5,000 liter bioreactor that's plastic, I can probably do away with everything except your 25,000 liter tank. So again, some positive opportunities there. As we go away from the facility itself and we start looking at technology, one of the things that I do want to talk about is batch versus continuous. Okay? So the continuous aspect of it is to do things over and over again as fast as you can, right? Run at steady state. I'm not going to get into the science or the engineering behind it, but there are incredible opportunities using perfusion bioreactors. You can get 5 to 20-fold higher. So then think back to that last scenario. If you were to run a perfusion N-1, you could inoculate your bioreactor with a much smaller tank. So you don't even need those big stainless steel N-1 seed bioreactors anymore. Now you go in with a much smaller tank, higher cell densities, lower volumes you inoculate with. So there's a lot of really good opportunities here. People use production bioreactors commercially. They work really well at using perfusion. They get very high cell culture, and they keep them for 35, 40 days. Again, contamination becomes an issue. The other issue with perfusion is the amount of media you use. So something to keep in mind when you're doing that. Just something that is a little harder to do is continuous chromatography. There is a way to do it that I call overload chromatography, where you fill up your column to 100%, and all the overload of that you put onto your second column, continue loading your second column while you elute your first column. So there is some opportunity there. Much harder to do in manufacturing, and not as many people are really investing in this yet. Give it time. I can see, or I've seen in different presentations, 20 to 100% productivity gains. So cheaper resin, better utility of your resins. This is just a graph of, I mentioned some things have changed over time. Cell culture titers are one of those. And here's a 30-year graph or so from when I first started to now. And you can see almost an order of magnitude change. And I've seen lots of productions now that are in the 10, 15 gram per liter range. So there are some huge opportunities here. This is really just a advertisement slide, if you will, of all the different types of chromatography resins that are out there. And on the right-hand side, you see there have been increases in chromatography resins, and this example here is showing capacity of protein A, which is the amount of protein I can put on a resin at one time. And again, you're looking at doublings and triplings on just your primary capture column. The other thing that's interesting to me is the data analytics. Again, I mentioned before I'd like to be able to sit in my computer. The only way I can do that is if all my data is there. And that's what this data lake is showing. And remember this, because the next thing I'm doing is going to talk about Industry 4.1, and that data lake is primary to that. So, where are we today? This graph up here is, I took this, just a generic Google file, but that's what I would consider as Industry 4.0, right? So the first generation, really that's our steam engines, right? That's the turn of the 19th century, or the 1900s. We get into mass production, thank you, Henry Ford. We get into some computer and automation. And then fourth, we don't touch anything, everything is done automated. So that's Industry 4.0. I could ask, if we had more time, we could go around and say, where do you think we are in biotech? But I'm gonna tell you, we're not even here yet. So the aerotech industry, some of these other industries, they're pumping out these things. We're struggling between Henry Ford and Bill Gates, right? So there's still a lot of room here. One of the nice things about Industry 4.0 is this use of artificial intelligence and the internet, the industrial Internet of Things, sorry, the IIoT, which again is to have everything available at your fingertips. Okay. The other thing that I like is this idea of a digital twin. So think of in your laboratory, you're doing your experiments, you make all your experiments, you make a model, and then that's the last time you have to do anything. After that, everything is done through a computer. That's the idea behind this digital twin. Or if you look at it from manufacturing, I have enough data on my manufacturing process, or I can make a model, and that model will be valid. So if I need to have a change, if I need to talk about a deviation, I can use that digital twin to do that. Okay. So where are we today? I explained a little bit, where do I think we're going in the future? And again, this is just my opinion. It could be right, it could be wrong. You know, bring me back in 10 years and you can throw rotten tomatoes at me. So I do think we're going away from large proteins into specialized medicines. Dr. Temple's presentation is a great example of that. I do think we will still need the 20,000 liter tanks because there are going to be systemic ailments out there. Rheumatoid arthritis comes to mind. I haven't seen a cure for that yet. I see masking agents. Until someone can cure it, maybe that's one of the things Dr. Temple will work on next, we're going to need big tanks. I don't think we'll see a lot of tanks being built in the future, a lot of big facilities, but they'll still be built. I think in the future we're going to go to those smaller facilities with a lot of either hybrid technologies or all disposable. And I mentioned the cell and gene therapy stuff. I would like the bottom thing to come true because I do want to, I'm an engineer. Engineers are lazy by trade. I don't want to work a lot. I want to have things done on a computer. But I don't think I'm actually going to see Industry 4.0 while I'm here. Hopefully you guys will take this forward. And with that, I think I'm done. Yep. So thank you very much. I think I have time for one question.
Thank you, Dr. Bates. Any questions from audience? One question from the online. I want to mutate Alexis. Go for it.
Hi, my name's Alexis. I'm
Please
from
come,
here.
Alexis, come to the microphone for the recording. There you go. Don't be shy.
I'm a little short, so hopefully you can hear me.
That's okay.
I'm a fifth year in the Molecular Biosciences program. I've had a couple internships in the industry, and I've seen a push or a trend towards, like you said, the smaller and multi-product plants where they're using a lot of the single-use technologies.
Yeah.
So I just appreciate that you talked about that. And then my question is, what are some bottlenecks you think that would come with transitioning towards automation?
Okay, that's good. Towards automation. So the biggest problem with automation is equipment. So we can talk all about how we want to automate everything, but the fact of the matter is a lot of our manufacturing facilities are built with older technologies, and they don't have the capability of doing the Internet of Things because they don't have good Wi-Fi in the buildings. Or when they were built, they ran electrical cord, but nobody thought to run, fiber optics. So that is really one of the downsides that I've seen. The other is automation is not easy. It's much easier to manually turn. And I think about like electronic batch records, things like that come to mind, running everything through a computer. It's really hard to do that time and time again. And then you mentioned facilities with multi-products. Every product has to have a different automation. So every step along the way, from purifying water to using your plant to cleaning your facility, not to mention all the different steps, they all have their own automation. And that's where it gets kind of complicated. Is it the right thing to do? Absolutely. Is it what's done? Sort of. But I would like to see it get more and more. I'm a realist too, and I know when there's somebody in finance telling you to save money and you're trying to spend $1 million on a new computer system, sometimes you get it, sometimes you don't. But that was a really good question. Thank you very much for that.
Thank you.
Well, we have questions online, but if you have time for another question.
Let's save it until the end. I think we're going to move on to the next talk, but let's thank Dr. Bates again for the excellent talk. So our final speaker for this part of the symposium is Professor Matthew Delisa. Dr. Delisa is the William L. Lewis Professor of Chemical and Biomolecular Engineering at Cornell University. He's also the director of the Cornell Institute of Biotechnology. And his research focuses on understanding and controlling the molecular mechanisms of protein biogenesis. Professor Delisa has received numerous awards, including election to the American Institute of Medical and Biological Engineering, the American Academy of Microbiology, and the American Association for the Advancement of Science. Today, Dr. Delisa will be sharing his group's work on cell-free approaches for biomanufacturing therapeutic glycoproteins and conjugate vaccines. It's my pleasure to introduce Dr. Matt Delisa.
Okay, great. Well, good afternoon, everybody. It's a pleasure to be here today, and I want to start by first thanking Kamal and Julian for the invitation to come here and participate in this symposium to kick off the Stack Family Center for Biomanufacturing. I had the great pleasure today, earlier, before coming here, to actually tour the facility, and it made me want to become a student again and go back to hands-on training with equipment of that quality and an opportunity and an environment for making things like the Industry 4.0 for biotech a reality. So we're looking forward to big things from you guys. We're not too far up the road at Cornell University and are very excited in looking for ways to connect with the center here and advance some of the ideas and some of the training and the opportunities, most importantly for students who are entering into this really exciting time in biotech and biopharma. So today, as Julian mentioned, I'm going to spend the time that I have telling you about some work that's been going on in my lab for the last 6 or 7 years on cell-free approaches to biomanufacturing. And the focus today is going to be on attempting to use these systems to make therapeutic glycoproteins and conjugate vaccines. And I want to thank our previous speakers, in particular Ron, for telling us about the current state of industry, where it's headed. And I'd like to hopefully have you leave today realizing that while cells are still the workhorse of the industry, there are opportunities to use the machinery inside of cells in the absence of cellular membranes to develop new manufacturing processes. And hopefully some of the things I'll share with you are an eye towards that future. So I want to begin by saying that the vast majority of human drugs that are proteins, so think monoclonal antibodies, but there are many others out there. The vast majority of these are modified with complex carbohydrates known as glycans. And the process by which they're modified is known as glycosylation. And we know these modifications are really, really important for the activity, the stability, the three-dimensional structure, the efficacy of the drugs. And so making drugs that are not just proteins, but that are modified with proper carbohydrate modifications is a really critical part of biomanufacturing. And in fact, the more complicated drugs get, the more attention we have to pay to these types of modifications. There are two major types of protein glycosylation that you should be familiar with in nature. There are many other types, but two major ones that I think are most relevant to the biotech industry. Those would be N-linked glycosylation, which is the attachment of a complex sugar known as a glycan to an asparagine residue in a protein, and O-linked glycosylation, which is the attachment of a glycan to a serine or a threonine residue. So if we think about protein drugs where these types of modifications are important, I already mentioned in the case of monoclonal antibodies, virtually all monoclonal antibodies carry a N-glycan in their Fc domains. In fact, they carry two. And these structures are absolutely crucial for the downstream activity of antibodies in terms of recruiting the immune system and eliminating foreign material and foreign invaders from our bodies. But there are many other types of glycoproteins out there, and as you can see from a few of the examples I have on the slide, they can be quite complicated. So the molecule on the left, that's EPO, so any cyclists in the audience? And if there are, I hope you're not using this molecule, but if you are, then shame on you. But this protein is used therapeutically and can have great benefit. And what makes it particularly challenging from a manufacturing perspective is that it's not just a protein, but it has 4 of these glycans attached. 3 of them are of this N-linked variety, and 1 is of the O-linked variety. So these can be quite complicated. And then the last example to the far right are what are known as conjugate vaccines. And in fact, some of the most effective vaccines on the planet for dealing with infectious bacteria are these types of conjugate vaccines, which are comprised of a protein carrier that's modified with an oligosaccharide. And that oligosaccharide is derived from the surface of the bacterial pathogen that you're trying to protect against. So these are incredibly robust and protective, but very challenging to make from a manufacturing perspective. And I'll talk a little about these later on as I go through the slides here. So in making any glycoprotein, so if you're interested in manufacturing a monoclonal antibody and you want to use a CHO cell, the good news is these cells are in fact used primarily because they have machinery that introduces these very complicated carbohydrate modifications. If you look under the hood of a CHO cell to try and understand exactly how these cells perform these processes, I don't have time to go into all of the exquisite details, but shown here in this cartoon, and it's, I apologize, quite small here, but the takeaway is that it's really, really complicated. There are dozens of enzymes that are involved in this process. It spans multiple subcellular compartments, from the cytosol to the endoplasmic reticulum, to the early Golgi, to the late Golgi, and then usually in the case of antibodies and other protein drugs, they're secreted out of the cell. So it's a very complicated process. When it's done right, it is very effective, but very challenging to control and to get it right. And so this has led many, both in industry and in academia, to try and find better ways of controlling and introducing complex carbohydrate structures onto proteins. And so my lab has been very interested in trying to find some of these alternatives. And one of the things that we've looked to is, as I mentioned at the very beginning, cell-free technologies. Now, cell-free systems, cell-free protein synthesis in general, is a very long-standing technology. It's been around for a very long time. For making recombinant proteins, it's quite powerful for a number of reasons, and I've highlighted a few of those here. First and foremost, what it allows you to do is to decouple the synthesis of your catalysts, so all of the protein synthesis machinery can be decoupled from their utilization. So you can make all the catalysts when you grow your cells and then prepare the cell-free lysate, but then you can store that lysate and use it at any time in the future. And when you use it in the future, now your only focus is on using those catalysts for carrying out the functions of making protein. So these two important steps can be decoupled. And really what that means is that by moving outside of a cell, you're no longer constrained by a physical or biological barrier, the cell membrane, and you're also no longer subject to cell viability constraints. When we set up these massive stainless steel bioreactors or disposable bioreactors, which are also quite large, as we just saw in Ron's presentation, one of the main challenges is we're trying to keep something alive inside those reactors. And productivity is intimately linked to keeping whatever it is inside that reactor alive and healthy. And oftentimes, what we're asking those cells to do, which is to make massive quantities of our protein products, of our monoclonal antibody, oftentimes asking cells to do that makes cells unhappy, right? And they start to push back. And viability is usually the expense, right? That cell death is always something that we're pushing against. So it's a tug of war between making more product, but keeping our catalyst system, our cell, alive. So cell-free systems are powerful because now that constraint is completely lifted. And so if you've thought about or even done anything with cell-free protein synthesis in your lifetime, you're probably thinking of really small microcentrifuge tubes, which is still where most of the work is being done, including a lot of the experiments I'm going to share with you today. In the laboratory, we're operating in very small volumes. However, in the last, I would say, 10 years or so, there has been a technical renaissance in cell-free systems, and many labs coming to operate in this domain. And that renaissance has led to a pretty significant scaling of this technology. There are now several startup companies that are growing and becoming quite successful. Sutro Biopharma, Swift Scale Biologics. And these companies are getting these small microcentrifuge tubes of cell-free reactions scaled to 300 liters, even 1,000 liters in some cases. So we're not quite at the 10,000 liters, but they can be scaled to quite large systems despite how you may think of these systems. And they are compatible with a lot of the current infrastructure that we have. They can be operated, I've seen them operated in 300-liter stainless steel reactors. I've seen them operated in wave bioreactor in bags, disposable bags. So there's a real interesting opportunity and promise here with this technology. But the other beauty is that they're also quite small and nimble. So if you're not trying to produce a protein drug for the entire planet, like Regeneron and others are trying to do in the pandemic, but rather are trying to make a drug for a small patient population or even an individual, this technology, I think, holds tremendous promise for that kind of opportunity. The future, one future of biomanufacturing 4.0 could actually be you walk into a pharmacy and the pharmacist makes the drug for you. Or you're in the hospital and your drug is manufactured in the room next to your hospital bed. Again, this is way out there. I don't think, I'd love to see this in my lifetime, but I do think we have to think about the biotech industry spanning many length scales. And I think a nice thing about cell-free systems is they have an opportunity to provide drug products across these pretty massive length scales in terms of manufacturing. Okay, so a simpler question that we have set out to address over the last few years in my lab is it possible to develop cell-free systems that incorporate these protein glycosylation reactions? So what I didn't say in the previous slide is that the other thing you may know about these cell-free systems is that the vast majority of them, and the ones that have been developed to these very large scales, are generally all based on extracts derived from bacterial cells, in particular E. coli. And as many of you may know, or hopefully if you take nothing else away from today, you will remember that E. coli cells naturally do not perform protein glycosylation. So those really important modifications can't be made by an E. coli host, but also can't be made in most cell-free lysates. So we thought this might be an opportunity for us to make a contribution. We had been working on installing protein glycosylation systems into E. coli bacteria, and that led us to think about perhaps developing lysates from those bacteria that would be enriched in the machinery necessary for installing complex carbohydrates onto proteins. We've tackled this problem at a high level, thinking about it in two distinct stages. So the first stage we're thinking about are the early steps of protein glycosylation. So the initial installation of some complex carbohydrate structure or precursor, or primer, if you will, onto a protein. And if we can succeed in doing that, in a second step, we can carry out in a cell-free environment glycosylation maturation reactions where we can tailor the glycan structures to look exactly like the ones we want that maybe are found on naturally occurring human antibodies, or could be glycoengineered to be structures that afford new properties to the biomolecule, which is a very exciting and fast-growing field. So today what I'm going to tell you about very quickly is on the left the work that we've been doing towards glycosylation initiation. So in essence, what we're trying to do here is take the guts from a E. coli cell that is carrying protein glycosylation machinery, use that to create cell-free extract that will, in most cases, be stored in a microcentrifuge tube and used to drive protein, glycoprotein synthesis simply by the addition of a DNA plasmid that encodes our product of interest. And if you do this right, you drop in your plasmid, you come back in less than a day, and your protein product is made with the desired carbohydrate modification directly on the protein. So your glycoprotein is now made in a very fast and convenient way. So this was work in my lab that was largely the effort of a really amazing graduate student, Tommy Jaro Antimichai. And so I don't have time to go into the details. The work is published, but he took glycoengineered E. coli in our lab, made lysate from those E. coli, and showed that you could generate a single pot system that was able to make a wide spectrum of different protein products bearing specific carbohydrate or glycan structures. So here are just a few examples, and again, don't be scared off by the Western blots, just the data to show you that in fact we have been able to make single chain antibodies, we've been able to make human EPO that I talked about previously, all with glycan modifications onto these proteins. And in the case of EPO specifically, not only were we able to make these as glycosylated proteins, but we showed in follow-on experiments that the molecules we were making were as active as commercially purchased EPO proteins. And again, we acquired those in a legal way, not from any cyclist in our lab. Okay, so we've done this with N-linked glycosylation. Aravind Natarajan in my lab came along and became interested in O-linked glycosylation. He engineered bacterial cells to carry out this type of protein modification and then adapted it for cell-free systems, and also had similar success demonstrating the attachment of O-linked sugars onto proteins of interest. The other thing that we've been able to do over the work of these two students that I just mentioned is we've been able to install a large array of different oligosaccharide structures onto proteins. And this fortuitously led us to an interesting observation that one of the oligosaccharides we were able to attach is not the kind you would see on a therapeutic protein, but is one of these surface oligosaccharides that's present on any type of bacteria, including bacterial pathogens. And so this led us to ask, in addition to making therapeutic glycoproteins, could we use the same technology to install bacterial long-chain oligosaccharides onto proteins as an entirely new route to making conjugate vaccines? And so the general idea here, and this is work that our lab did together with Mike Jewitzer at Northwestern and a really talented student there, Jessica Stark, they had this idea that we could, again, use all of the same concepts that I just shared with you, but what would be really powerful here is that these reaction systems, the lysates you make, can be lyophilized or freeze-dried. So you don't have to use them right away, right? And so then, in a freeze-dried format, these can be transported anywhere in the world. And you can do that without the need for a cold chain. So there's an opportunity here to think about entirely new paradigms for vaccine distribution and manufacture that overturns some of the major challenges we have in maintaining the cold chain, and then may even give you an opportunity to think about entirely major decentralized strategies for producing your product, on-demand or point-of-care protein manufacture. So these are some of the ideas that we thought could be addressed with a technology like this. So I don't have time to go into all the results, but let me just say that The technology has given rise to a very potent platform for making these conjugates. We estimate that the cost, so another beautiful thing is that this can be a very low-cost technology. These vaccine doses we're making come in at around about $5 per dose, which is still too high if you talk to folks at the Gates Foundation and elsewhere, but we're still way cheaper than the conventional technology, which runs about $200 per dose. So we think we're on the right track to not only creating a new technology, but also one that could be affordable and provide greater access to much needed vaccines. We have seen also, as I showed in the EPO example, that not only can we make these things, but they're efficacious. And this is an animal study that we did with some of these cell-free derived vaccine candidates, showing that they are able to completely protect a mouse from pathogen challenge, where the pathogen that was used in this experiment corresponded to the oligosaccharide structure that was attached to our protein carrier conjugate. So these are the two platforms. I told you about the one on the left. I don't have time today to tell you about how we take primer glycans that are initially installed on proteins and then turn them into whatever structures we want. But there's a huge opportunity to elaborate these glycans, opening up new opportunities for glycoengineering, which is basically a way of making biosuperior drugs, drugs that have better properties than their parental molecule because the carbohydrate structures themselves have been tweaked or modified to give a better therapeutic effect, something we're very interested on right now in my lab. So in summary, I'll just wrap up with this slide that highlights some of the things that I've already said. But really the last point is the one that I want to just reiterate, and that is we think there's a growing and pretty massive opportunity for cell-free technology, not to replace any of the things we've heard about today or any of the things that are going on here in the Biomanufacturing Center, but to operate alongside and provide alternatives. And that's really what we need, I think, as an industry, right, is to have lots of different shots on goal with lots of different technologies that can all be brought to bear in making the medicines that we need going forward. So hopefully the next time I come to the center, we'll have some cell-free systems that are up and running in those same bioreactors. And if not, I'll do my best to try and make that happen. So with that, let me thank first all of the great individuals, both students, postdocs, and collaborators that I've had the great fortune to work with on some of the things that I shared with you here today, the funding agencies that made a lot of the work possible. Thank everybody for your attention, and if there's time for a question or two, I'd be happy to take those.
Thank you, Dr. Delisa. Any questions from the audience? Online, we have actually a follow-up question for Dr. Bates, not for Dr. Delisa, so you can go back. Sure, please.
Matt, this is fantastic, so I greatly appreciate it. Getting back to your conjugated vaccines, so one of the questions that I would have is timeline. How long does it take from inception to having a working vaccine? Not necessarily through all the health authorities, not counting that.
Yeah, so great question. So we, broadly speaking, and not just for the conjugate vaccines, we think one of the real game-changing aspects of this cell-free technology is that when you have the lysates prepared and stored and ready to go, the timeline for making drug products. So you mentioned this in your talk about there's really a key race to get from discovery, early stage discovery, and getting small doses to the clinic, right? We think that's a real opportunity here with the technology. It's quite fast. From the time you identify a pathogen for a conjugate vaccine, it's quite fast because you actually clone the genetic pathway that encodes the oligosaccharide biosynthesis. So that's a day or two. You put it into the system probably takes less than a day, but let's just call it a day, 2 days max to make your product, depending on how much you're trying to make. So you could conceivably, we've done back of the envelope estimates, get this done in about a week if you're really turbocharged and you know what you're doing. And so with that in mind, we've been approached about pandemic response, right? And not just thinking about coronavirus, but all kinds of outbreaks that we've dealt with over the last any number of years. And so a technology like this we think has that opportunity. And the good news is you never actually have to handle the pathogen. Current conjugate vaccines, for example, you're handling the pathogen to shear off or remove those oligosaccharides. We never have to touch those. Everything is completely synthetic or biosynthetic. So it also, we think, creates a much safer work environment with less hazards in terms of material handling.
Yeah, that's fantastic. Thank you.
Sure.
Thank you, Dr. Delisa. Any other questions?
So I think we've picked up a little time along the way. So if there were any final questions for Ron, either we could entertain one of those. I believe one came in online.
Yes, there is a follow-up question from my former boss. Means I have to read it. Disposable reactors sounds like a very good idea for the industry. How do you think we can still introduce this to our students as we are educating the future for workforce of the industry?
So let me make sure I understood this. How can we introduce disposable reactor technology to students?
Or for downstream in our educational labs. And non-convex food, non-lab, because prefer new, brand new, we have XLR, we have some disposable like ATF from Retina. But I believe all the labs are really based on all these things.
Yeah, I mean, I think, so one of my slides I talked about cost, and it is easier for, you know, a university to work in stainless steel because they can often get a stainless steel bioreactor donated by a Bristol-Myers Squibb or glass even. A lot of the bioreactors in your labs are probably glass, not stainless steel, but regardless, non-disposable type. You know, the question is going to be cost. To get into the technology is the same. So whether it's glass or plastic, the technology and how to grow cells is the same. Matt's trying to get rid of that technology, but that's for a different discussion for a different day. But otherwise, it's really the same technology, and it's the same technology we've been using for decades and probably more. Same technology they use for beer, but just for a different substrate, right? But for the question at the table, I would say, you know, the disposable chromatography columns are going to be even harder because they're really, really expensive, and you can do a ton of chromatography in a week. So I think, you know, the important thing would be to show for centers and for academic labs or even industrial labs that don't have the capability to buy a lot of stuff, it would be just to show to demonstrate the comparability between the two techniques. Use your current technique, but make sure everyone's aware that there are other ways of doing the same thing. I don't think I would try and go out and buy, you know, disposable systems, because you're going to have to buy bags every time. You're going to have to buy tubes every time, connectors. And with chromatography columns, every experiment could be a new run. And these small columns, which are, you know, no bigger than a cigar, they're going to cost you $1,000. So, you know, think of a very good student. A chromatography run takes 4 hours, let's say. You know, they're running if it's a grad student, 3-4 a day, you know, because they don't sleep. But nonetheless, you're going to run out of a budget really fast. So that's where I would switch and just say, you don't have to use the right technologies. You just have to make the students aware and the investigators aware that there are other technologies being used. Maybe you have one system that uses that other technology as a demonstration system, and then you have, you know, the workhorses do everything else.
And packing our own columns, does that really count as, you know, if we pack them in plastic or something that you can see through, like glass, does that really count to introducing, do you think, the single-use technology? Still, we have to amend it with some kind of theory.
So the difference between packing, so the difference between packing and buying is really that. When you use a disposable column, it comes ready to go. You think of an HPLC column. We've all used HPLC columns, right? But think of throwing that away after every use. That's the difference. The packing a column yourself is an art. I mean, you could get people who know how to pack, and if you're in a certain lab, they're the people that everyone goes to. Can you pack this column? I really need it packed well, or whatever, right? Same thing in industry. We all have our people who can pack really well. On manufacturing floor, when we're talking about 80 centimeter, 1 meter, 2 meter columns, we get into more technologies that pack for you, but there always seems to be that one or two operators that just knows just the right packing velocity or just the right time to shut it off. That's just what it is. That is what you lose when you go to disposable. Is that good or bad? That's for a different discussion, a different day. But that's why I think when you're using disposables, you use it for that simplicity. Students in the lab, I think it's probably better to learn how to pack yourself. And there's a number of reasons why I would say, one of which is because you don't, like anything, do you learn when you succeed? No. You learn when you fail. And let me tell you, if you're packing your own columns, you're going to fail. And you're going to learn from that. And then you're going to realize, I overpacked this one, I underpacked this one. So that's why I would think, same thing with cell growth and any kind of reactors. You don't, you only learn when you get contaminations or your reactors go haywire, right? That's why there's always an opportunity there. But I think what I would recommend, former boss or current boss or whoever, is that you get a system to introduce the technologies to people, and then you go through the cheaper technologies or the cheaper usage of the same technology for everyday use.
Yeah,
Thank you very much.
That's a very good point, Ron. So column packing is very popular, and I think John Balchunas, can attest to that because they offer column packing as a training program. So this might be an idea for you to start a column packing one-day program. It will be desirable.
Absolutely. We have our hands on a industry expert, but she has given us prices, as you know, above average, because column packing, like you said, is an art, but we'll push for it. Thank you very much.
Thank you all for the great questions, both from online and in person. This has been a great session, so thanks for all the participation. And I'd like to extend a special thanks to our speakers, both in person and Dr. Temple, who joined us remotely. We appreciate their time joining us this afternoon. It was an honor to host them at the center this morning, and we're very glad that both Ron and Matt will be able to stick around for the panel. Dr. Rashid has put together a wonderful panel on workforce development this afternoon, so we are going to now take a 15-minute break, and there will be time to ask questions directly to the speakers during that break if you have any remaining inquiries, and we'll see everyone back around 2:45 for the panel. Thank you.
collaborate in helping with these new technologies and workforce that is needed and in demand by these biomanufacturing companies. So we have a great group with us today, and some are online and some are on-site. I'd like to start out with pictures I see on the screen. So right now I see Dr. Ali Demirci. Dr. Ali Demirci is a professor at Penn State University. Welcome aboard, Dr. Demirci. Thank you for joining us. I'm just going to go about introducing the name and where they come from, and then I'm going to ask everyone to introduce themselves in a more detailed way, what they do, and then you can know more about them and their activities. And we have some really big names here that they can really be a factor in the progress of this industry and the development of the workforce. So let's start with you, Dr. Demirci. Just tell us about yourself. You have 2-3 minutes to talk about whatever you want.
Well, thank you, Dr. Kamal, for inviting me, and also congratulating to everyone involved in developing and creating this great center. I hope, and I'm sure it will be a very successful one, serving not for academia, but also to industry, definitely. Well, I'm a professor in agricultural and biological engineering, and I'm also in charge of the CSL Bearing Fermentation Facility, which is a core facility open for researchers on campus and off-campus users that provides the large-scale fermentation. So I do research in fermentation and teach around the same area. So I'm very happy to be on this panel today to share my experience as much as needed. Thank you.
Thank you very much. Thank you for joining us, and we appreciate your time. Next, I'd like to introduce Dr. Andreas Kastan. Dr. Kastan is a strategic technology and business development leader for Cytiva. And Cytiva is very well represented in our center. We have a lot of equipment from Cytiva. So, Dr. Kastan, your microphone is yours.
Yeah, thank you very much. It's a great pleasure to be on this panel discussion, and I would have loved to be there in person as well, to see the center and interact with you in person. I'm with Cytiva, as said. I've been with that company now for 10 years. And prior to joining Cytiva, I've been in the biotech industry in Sweden, leading the upstream development and pilot manufacturing in a company called Swedish Orphan Biovitrum. Among other things, we were producing or are producing the factor VIII supply for Pfizer. But at Cytiva, I bring very much my experience from the industry into the company, and facilitate the development of products that help to meet the customer needs around cell culture. And I'm also leading a lot of the activities where we do application work with our products to basically inspire customers to develop better processes. So it's a pleasure to be here today.
And thank you for joining us all the way from Europe. How does the weather look like there?
Oh, it's very nice today, but it's getting colder now.
It's getting colder. We think we're going to have a thunderstorm, but haven't happened yet, I think. Okay, next I'd like to introduce Dr. Kinnisaku. Dr. Sakun is the Director of Virology at Antalya. Dr. Sakun, the microphone is yours. Thank you very much.
Hi, everybody. Thank you for the invite today. Again, I wish I was there too, so hopefully sometime in the future I can come visit and see everything in person. Yes, I'm the Director of Virology at Zeptometrics. We're over in Buffalo, New York. We basically are an infectious disease biotechnology company. I've been here for 16 years myself. We do have 4 main scientific departments, which are virology, bacteriology, immunology, and molecular biology. And we all interact to develop different products for the market. The main market that's been driving the growth of our company has been the diagnostic market. as far as we partner with a lot of those companies developing infectious disease assays. So you can imagine with COVID rampant right now, we've been extremely busy. We continue to grow, and we were actually just purchased by Antilia at the beginning of the year. So we're still in the process of kind of onboarding into that giant corporation. So it's a pleasure to be here today. Thank you.
Thank you very much for joining us. We really appreciate your time. Next, I'm going to go around the table. We probably know most of them from the presentations, the beautiful presentations that they gave, but maybe we can go through and then you want to say whatever, in addition to the introduction that Julian said. You have 2-3 minutes to say whatever you want.
Okay, well, I don't want to hog any time. I'll leave it for my colleagues here, but Matt Delisa, I'm a professor of chemical and biomolecular engineering at Cornell and also serve as the director of Cornell's Institute of Biotechnology. And again, I just want to thank everybody for the warm welcome and the invite, and looking forward to a lively discussion.
Okay.
All right, sure. So I'm Derek Brand. I'm the Chief Operating Officer at New York Bio. A little bit of a background on our group. We're the trades association for the biotech industry across the state of New York. We're a 501c6 organization, which means we do represent the industry in front of Albany. And we also serve to basically raise the profile of the industry, provide connections, and a whole number of other benefits to our member companies. So we have more than 200 member companies across the state. And over the last few years, we've kind of instituted a few new initiatives. One of them is we're actually really working to get closer to patients. We held our first ever patient engagement summit this past spring. We've also worked to bring the biotech industry much closer to the financial industry and the public markets. And we've done a few emerging company showcases partnered with the New York Stock Exchange. Coming up on September 27th, we have our annual meeting. We're happy that we're actually able to do part of it in person. And of course, it'll be hybrid, so we'll have a virtual option as well. For everybody that's both here and online, we do have a discount code on registration that we're very happy to extend to people. So I believe the folks will put into the chat both a link to the registration and a link to that discount code. So we hope you can join us. Honestly, New York over the past few years have seen just tremendous growth in the biotech industry. I love the fact that I could come out here today. Thank you so much for the tour this morning. And I get excited to see really great things like this pop up more and more often. And it just makes me even more positive that New York is a great place for biotech.
Thank you, Derek. It's really a great pleasure to have Derek with us because I think there's a lot of synergy between our center and what we do representing the New York Bio. We're really hoping that we're going to be working together more and more, and we are a member, but we're going to renew the memberships because it's really important. The bio organization in each state are playing a great role in advancing biotechnology, and so thank you for driving up, and thank you for being part of this panel discussion. Now I'm going to turn to John Balchunas. Mr. Balchunas is not new for the workforce development efforts. He is the Assistant Director of the BTEC. Don't bother me. He's the Assistant Director of the Industrial Programs at BTEC. BTEC is the Biotechnology Training and Education Center. These words are becoming around, you know. It started out with them, and then at Worcester Polytech, they said, well, they are BTEC, we're going to be BETC, and now we are CBET, and so it's all focused on education and training. But in addition to his post at NC State University's BTEC, I'm very pleased to have John joining us because he represents the workforce. He's the director of the workforce at NIMBLE, the National Institute for Innovations in Manufacturing Biologics. And so I'm happy that you are here, and please tell us a little bit more about you and what you do.
Yeah, well, and I appreciate the invite to come up from North Carolina, you know, to be part of this panel. So I think, and I look forward to seeing the center after the session as well. I didn't get a chance to see it this morning. So I guess as Kamal said, I wear two different hats. One hat is I am the workforce director for NIMBLE, the National Institute for Innovation in Manufacturing Biopharmaceuticals. Long story short, we're a big federally funded public-private partnership. We're a collection of academic institutions of which Albany College of Pharmacy and Health Science is a member. A bunch of large pharma members, a bunch of community colleges. We fund projects and we do things. Basically, we're there to drive innovation both on the technology side as well as on the workforce side. So that's one hat. The other hat, is grounded back in North Carolina. As Kamal mentioned, I lead the industry training program or the professional development program at BTEC, the Biomanufacturing Training and Education Center. The BTEC, I always, I can humbly say BTEC was the first of our kind, the first of these centers really in the world. We opened back in 2007. I think what's relevant to this conversation more so is honestly, North Carolina has done, and I'm very humbled to be from North Carolina, who has done a very, very beautiful job at attracting industry to the state or attracting biomanufacturing to the state through, education, training, and workforce development initiatives. And I mean, if anybody's been watching the news over the last year, just about every single biomanufacturing site, it seems like, is landing and locating to North Carolina. It's raising a new issue for us in terms of how we're going to get them all trained. So, how we can build that workforce. So,
thank you for
anyway, glad to be here.
All the way from North Carolina. We appreciate that.
Happy to be here.
Next is our good friend. I think Ron just introduced himself. Dr. Bates is a very good friend of the center. And we had the pleasure of having him helping us out with the center development in our webcast. Dr. Matt Delisa was also a speaker in our program that we offered webinars to introduce the center to the bio-based industry. So Ron is really great help for us, in our training programs. He is teaching in the training programs. And we want you to continue and add more to that for help. The center needs all the help from our friends in industry. So I know you were introduced, but if you want to add any other words, please, you go ahead.
Hi, my name is Ron Bates. I'm currently working at a company called Immunovant, and we have offices in New York City and North Carolina. Go figure. So I listened to what John was saying. Totally understand that place is blowing up. So, but for my career, the one thing that I've always done is needed a very strong workforce from whichever company I was working at the time. So a center like this is and also the different centers that Camille mentioned in Worcester and North Carolina and a couple other places are tremendously valuable. And for you students out there, do realize that they are great opportunities to learn, so please take all advantage of that. That's about it, Camille. Go ahead.
Thank you very much. Thank you for being there for us. We appreciate your time. I think the way we will go about this is that we're going to start with some questions here so that we can start the discussion going. Then we're going to open up to you. If you have a question, you can ask, and the same way you come to the microphone, and then if the question is not clear, I can repeat it so that we can hear it very well. So I'd like to start the discussion by asking one question that is on my mind. I mean, there are a lot of centers, like John said, and then there are many other centers besides this BET, CBET. There are centers in Texas and in Georgia, and Maryland used to have one, and so on. But how come, there are, and that's maybe some question that John, you may, because you're both representing academia and you are with this Nimble Institute. There are some tendency in academia that say that, for example, training is something that industry do in-house. And I know, Ron, you can pitch in, with the same question. Some academic people have difficulty, digesting that we're doing workforce development in an academic institution. And the question is, why do we have to do that, Because these industries, they have their own on-site training. When a new employee goes there, they train them. So why do we have to do this training when they are doing it? And I think from your perspective, John, you have been involved with BTEC for over 15 years, and now with Nimble for over 5 years. We had the pleasure working together. I ask you, with all this, investments universities are doing around the country, establishing centers, and with Nimble's putting millions of dollars in funding academic institutions to do workforce for industry, how would you answer those reluctant academic people who say that workforce development does not belong on academic campuses?
It's a tricky question to answer, honestly, because I think most of the institutions that I'm familiar with, it would be like preaching to the choir. I think a lot of the institutions that I'm aware of, they get it, and so there's not as many reluctant faculty. That said, I can change the question a little bit, or change my answer a little bit to say that one challenge that I think is not unique to any of the centers is finding or teaching academic faculty how to teach to industry, or how to teach in an applied way. And I think it's becoming even more critical now for all these centers, and it's a shared probably issue for everybody. Back when BTech opened, and probably for the first 10 years, I'd say 99, if not 100% of our staff were from industry. I think with the extreme growth within the industry now, I think there's a lot of competition. Basically there's a lot of jobs that people that would normally come from industry to come teach at these centers are now back in industry. And so I think there's a whole new reason to probably motivate faculty to get engaged in teaching in more of an applied way. But generally, I think a lot of the institutions that are doing this kind of training are coming from more of an applied mentality to start with, and they're used to having those connections with industry.
Maybe you want to chip in because you represent the biomanufacturing industry.
Yeah, I would look at it as a couple of different ways. I mean, so, where does workforce training fit into academia? There's a couple ways to look at it. I mean, one of the things I think of, in what John was talking about too, I know a lot of people in industry who are really good, really smart, probably teach some of these courses, but they're not the best teachers either. I mean, the fundamentals are the same, whether or not they're taught by someone who spent their career in industry or someone who's spent part of their time in academia or all their time in academia. So the difficult thing with workforce training is making sure that the trainer is actually qualified. So I mean, the first thing I would say to that question is, where better would it go? I mean, so I can turn that around a little bit and start talking about on-the-job training of college students, internships,
absolutely,
sabbaticals, things like that. And those are all fantastic. I have used them throughout my career. I have hired people to do interns, co-ops, and then hired them full-time. But once they become full-time, what do we do? And that's where I think the relationship with some of these consortiums, BTech is one, what we're talking about here, any of these opportunities is fantastic, because it doesn't, when you get your degree, I know you guys sing to this choir too, but when you get your degree, the learning doesn't stop. And, I can provide all the on-the-job training I want, but I'm not going to teach someone who doesn't understand cell culture. I don't have the time for that. But they can get a feel for that by going to take some of these courses. And the idea of going and being able to, feel, see, touch is so much more than, reading a textbook. So that's where I think the link is between industry and academic. I mean, we've all heard through the years people go back after they start working and they get an advanced degree. I can tell you most of those advanced degrees are MBAs, and they're utterly worthless. I mean, they look good, don't get me wrong, and if you have an MBA program here, I'm sure it's fantastic. But they don't really help people do their job, right? They help them get their next job. And I don't think our headhunters here yet, but nonetheless, that's not my concern. My concern is, how do I make you better at this job, at your job? And these hands-on training courses, they're the best. And if I can do it in conjunction with a proven entity like a university or like a training facility, that's where I would want to go.
Thank you. And actually, you brought up a very good point that I want to go to Dr. Demerci. Dr. Demerci, I want to ask you, because Ron brought up the point of sabbaticals, faculty sabbaticals at industry. And I know you have done a couple of them. You did a sabbatical at Merck, and then you did a sabbatical at CSL fermentation facility in Europe. Can you tell us a little bit about how useful was that and how that led into more interaction between those companies that you had the sabbatical with and Penn State as a university, and also you yourself being more focused on preparing your students for positions in industry, knowing what the industry needs?
Well, thank you for the opportunity. I completely agree what it was said just a minute ago. Training and workforce development is very critical for the industry, and it has to be done actually both by academic institutions as well as by the industry itself. So it will like a chicken and egg kind of story. But this is even more than that, because we have to help each other to learn the best way of educating. In academia, we may think we know everything, what industry knows about the fundamentals and etc. But it's wrong. Industry can tell us exactly what applications is important for them, so we can educate the students accordingly. So as an academic researcher, of course I teach in the classroom day to day, I have no industrial experience at all. Of course we do field trips, of course we do visits to visit the plants and etc. But it's good, it's like a touristic visit, you can learn a little bit, but it is not the same thing, you work there day to day. So universities give us opportunity every 6-7 years to take a sabbatical and go somewhere and improve yourself and come back and be helpful to your students and bring some ideas back to university. So I had a chance to do that twice, and of course I could go to another academic institutions and make a couple publications that will look my CV better, but personally, I prefer to go to work for an industry. So in 2007, I took that opportunity and I went and worked at Merck. It was their R&D facility. I wanted to know how research is done at the industrial R&D center. So it gave me a lot of insights, unbelievable. And then just 2 years ago, before COVID hit, I had a chance to go my second sabbatical, and I did that in Europe, in Switzerland. I took the base in CSL Behring production plant. This time, I didn't want to go and do just one research project that I can improve on technique or whatever I'm working on. I wanted to know overall how a pharmaceutical industry works. So basically, in my 6 months of time, I visited everything from R&D, production, quality, marketing, regulations. It was mind-blowing for me as someone coming from academia to understand how complex the industry is. That's really, I cannot read and learn by myself just reading in my office. It gave me a first-hand opportunity to open up my eyes and get a little better understanding what kind of students we need to educate for the industry. And also I took that opportunity to ask questions from the top of the company all the way to the technicians. What do you expect that our students should be prepared for? Or what are they missing? So they gave me all their perspective. Of course, I all collect that information. And when I came back, I started changing a little bit my curriculum. And I explained to my colleague what they should pay attention, what they should also prepare themselves in addition to measure that they are working in. Like I heard in the previous presentation, Industry 4, in academia, we don't really hear about those. So now, with my experience, I push our program so that we now encourage our students to take minor, at least in Industry 4 related areas. Like we have a minor at Penn State, Six Sigma. So now our students are encouraged to take that minor so they can understand the Industry 4, and when they start their professional jobs, they'll be mindful of that and maybe improve what the process they are doing there. So my suggestion to all academic institutions, with the help of the industry, I think they should open up their door and accept the faculty as a sabbatical, 6 months or up to a year. Not only visit a week or two, like a day or two to host them, because it doesn't give you the same value. So I learn a lot. I now transfer that information to my students. I prepare them much better. And hopefully when they go back to first their jobs, they will be aware of all these challenges while they are still in the school. So I cannot encourage enough my colleague, and also I cannot enough encourage the industry partners to open up their doors so we can help each other learn better. We have the same goal, how we can make the next generation ready to pick up the challenges we have in the industry or other problems in the world. So I think these are the few things I can share personally, but if there is another question, of course I can provide more insight on that.
Thank you very much. That was very nice. And that brings me to Derek. You know, Derek, you are the big chief of New York Bio. And we talked about...
I'm technically the secondary chief. There is somebody out over.
Well, you are, you know what? But Derek, now listening to Dr. Demirci, they're talking about, you know, sabbatical, and then Ron talked about, you know, internship and fellowships and stuff like that. And as the representative of the bio-based industry in the state of New York, what is your thinking about how you can really encourage your base of the biotech companies in the state of New York to get opened up to academic institutions and to get more internships available for students at different academic institutions that they go and they can benefit from these students, and they may end up their employees because they work there for a couple of times and they trust them, they hire them. So what do you think that New York Bio can do with this regard in bringing academic institutions like Cornell University and ICPHS and other institutions, SUNY Poly, the others, how we can bring a network of these academic and industry together so that we can discuss this point and say how we go about, you know, how we go about interacting more and working together more and providing sabbaticals for the faculty, opening up the minds of the faculty of what's going on inside the industry, and also internship that would really lead into good employees for industry being based there, being tested there, and then they don't have to go look further. So what is your opinion on that?
Well, I mean, there's a lot in that, right? I think really what we're getting towards is kind of the opportunity for a center like this to provide the chance to basically hone collaborations with industry and for there to be kind of sharing of best practices, and really when you think about the experiences that the students will get here, having those experiences that are going to be kind of most relevant to things where they would step into an industry job. I mean, in thinking about Dr. Delisa's presentation, you think about, you know, the practical kind of applications that are now becoming much, much more commonplace in academia, where you are actually looking at manufacturing technologies, where you're looking at kind of pushing the envelope for really where the next wave of innovations in industry are going to come from, right? I mean, I think we've seen this in chemistry. We've seen this in so many places where biotechs will look to academia and venture capital will look to academia for where the next breakthroughs in industry are going to come from. In a lot of ways, the bioprocess industry has, you know, developed, you know, mostly within each of the companies that it's developed, and there's best practices, there's standards, because largely academia hasn't existed to manufacture things. And now that's changing a little bit because we're thinking about how do we make better vaccines? How do we really push the boundaries of the bioreactors and the outputs of these things to make up for some of the challenges that, you know, that industry is facing? You know, how do you produce vaccines or other therapeutics in, say, a developing world setting where, you know, doing it for $5 a dose isn't quite enough? Right? And it's these kind of answers that I think people can get great hands-on experience with here, and, you know, have that be widely applicable to, you know, making monoclonal antibodies, making next generation vaccines, making gene therapies, et cetera. It's those types of skills that I think are going to be adaptable and useful in just about any industry setting. So to kind of come back to your original question, I think what this represents is really a great opportunity for collaboration. I think each company probably does that a little differently. So I think some of it is going to depend on which companies kind of see the opportunity and kind of finding a partner where it's the right timing to work best together. From New York Bio's perspective, what we like to do is we like to make sure that our members, our academic institutions, and everyone, we like to raise that bar of connectivity. We like to get important events, important opportunities out there, and really try and make that whole network a little bit more efficient so that those partners can find each other a little easier. When it comes down to it, we want to see innovation advance. We want our companies and our members to have the best opportunities to be the best that we can. So it's programs like this where we basically say we'll dig in and do whatever we can to either help promote it, help design it if we have to, help kind of put people together in the room to talk about it at the early stages, so that as it's being built and as it grows, we really see the best benefit for really your institution, the partner institution, and ultimately the workforce that you're going to develop.
Thank you very much. Now I want to turn to Andreas. Andreas, you represent Cytiva. Cytiva is one of the best companies when we talk about suppliers for the biomanufacturing industry. You have done a lot of original research in developing these technologies, whether it's a single-use technology or optimizing bioreactor systems for cell culture fermentation, for purification and stuff like that. How do you see Cytiva's and alike, Thermo Fishers and Applicons and Eppendorfs and what have you, how do you see yourself collaborating with centers like us by really coming along and academic institutions are really short on money. So we build a center and we put a lot of quality equipment in it, but we will not be able to replace them next year or the year after that, but then there will be technologies developed by you. How would you see some collaboration from your side with these academic institutions, with Cornell New York Biotechnology Institute, where they can use these new technologies on a loaner, on a permanent loan, on a donation, perhaps? How do you see that Cytiva and alike companies become more involved? I know you have the fast track where you train industry in all aspects of biomanufacturing, upstream, downstream, but then you can have access to centers like ours where you can be a part of instructional team, and you can always be part of the delivery system. And sometimes when a new technology is out, it could be either brought in and out for an exercise, or it could be like a permanent loan, or it could be a donation. How do you feel about these?
Yeah, if I start answering about on a kind of high level, I think that there is a huge lack of trained staff in basically three areas. What I see right now in the digital space, people that know help to kind of handle data and make sense of data, in the manufacturing and in the R&D space, and I think all these areas need to be addressed. And if I look at the kind of places where this has worked successfully, from a European perspective, for example, in Ireland or in Switzerland, to some degree in Sweden as well, there is a very close interaction between the industry of providing staff basically on all levels. Just the example, Switzerland, I mean, everybody who has been there will see how expensive stuff is, and it's a little bit surprising how Switzerland can compete with other countries with much lower salaries. But the answer is really there's highly qualified work staff available for the manufacturing floor, for leading the development and developing new drugs and running plants. And this interaction is I think what is needed, that new stuff is trained and there is an exchange back and forth. And we as Cytiva, if I say how I personally work, I work with universities, for example, across the street here in Stockholm with the Royal Institute of Technology. They have a yearly course where students work for 6-8 weeks full-time in the lab and with the task of playing or setting up a company to produce an enzyme in an E. coli process. And they start from scratch and form a project team and a kind of project lead, split up in sub-teams, do process development, do computer simulations, set up an upstream and a downstream process, the analytics, and then finally in the last week they do their pilot run in 100-liter scale. And this is exactly the type of insights that the students get, which makes them really attractive for an industrial position, because they know what a yield in a process step means, and so on, beyond the kind of academic perspective. Apart from this, we as Cytiva interact a lot with universities to leverage the kind of knowledge, the depths in advanced analytics, mathematical modeling, and working together with different industries across Europe. And that means also that we place our equipment there and let the academic partner work with the equipment. And apart from the knowledge we gain, we also gain interesting application data which we can share with our customers, which might be more credible because it comes from a third party lab. So it's a lot of interaction that we have with different institutes. I mean, we are with Nimble, we are working with NIBRD, we are working with BOKO in Vienna, the Royal Institute of Technology in Stockholm, different universities across the country. So there is a kind of very vivid ecosystem of interactions where we always try to help the academic partner as much as possible when it comes to disposables or cell culture media to facilitate this work, but also with the insights we have about industrial processes, so that it's fruitful, I think, for all parties.
Thank you very much, but I want to ask you humbly, can you add CBET to your list? Thank you so much. I think, we have a lot of interaction with your group here, and we have a lot of your equipment in our, especially downstream lab. And so, I mean, collaboration, interaction, helping out with teaching, it will be a great way to start, and then you can mention with other universities that you're working with CBET at ACPHS.
Yeah, and it always starts with a problem statement, something you want to accomplish, and then kind of build up a momentum from that.
Okay. Thank you so much. Now I'm going to turn into another topic, and I want to bring Kelly into the discussion because she's the director of virology, and I know we're going to get into that subject with Kelly, and then I'm going to extend to Matt in discussing cell-free vaccine manufacturing probably. But Kelly, what is your feeling about, that as an expert in infectious diseases and working in the industry for so many years, what is your thinking about where we are heading? I mean, we lost Kelly. She did? Okay, yeah. So what do you think? You are a New York company and we are New York academic institutions. How can we collaborate? Because I think at CBET we're thinking about, we offered a workshop on vaccine manufacturing a few months ago, but we're thinking of developing a training program on vaccine manufacturing, and we're going to need your help. So where do you think that your company can be a collaborator with us, helping us out, with our mission, but then we can also help you with, student internships, and then also having some, industry academia collaboration, some translational research work that can be done with our faculty. How you go about that? And I'm going to ask a lot of questions, but you go ahead and answer whichever you want.
Okay, I'll probably touch on a couple of things that you've asked. So just again, as a little bit of background, we have a couple of biosafety level 3 labs as well as biosafety level 2 labs at Zeptometrics. So we're able to work hands-on with a number of different microorganisms. So obviously my department is more involved in the growth and processing of viruses. So kind of speaking historically, we've had relationships with some local colleges and even high schools where students can come in and just do, your basic run-of-the-mill type of internship. So if somebody's going to come in, they'll definitely learn aseptic technique. They'll learn how to manipulate cell lines. They'll learn how to infect and harvest viruses, testing, obviously personal protection equipment, and all those safety parameters go along with the training from whether I'm hiring somebody new or whether I'm bringing in an intern. And just to kind of, it's interesting to talk about workforce development at the academia level because, thinking back to when I was in grad school, it was frowned upon, if you were trying to get a higher degree and were planning to go into industry. And now I love the fact that you guys are taking the time to put into your students that which is going to make them a better worker in this field or whichever field that they decide to go into. Because from my perspective, when you're in academia, you have the luxury or the gift of time, right? But if you're in industry, you have the gift of money. So when you're kind of faced and bridging those two, one of the issues that I've had with some of the workforce is if they come from academia, they have time, right? You have time to think, you have time to plan, you have time to read articles, you have time to run experiments and ask questions. That's kind of like a slower rate of output. Not that I'm saying that's a bad thing, but if you're going to move that into the workforce where you need, manufacturing done, you need your cell lines taken care of, and you need to be prepared for those next steps of, whether you're doing some type of bioprocessing or whether you're planning for viral infections or you're making something specific for a customer, time is money. You have to be really good at your planning. You have to be on your game. There isn't necessarily all that time to put into reading and thinking and it's pretty much you have that output. So that rate of output is much higher once you're in industry. So I think if you kind of take that and think about it and the students think about it, so when you're preparing these students, you're kind of bridging both of those. So you want to put the time in to develop experiments and questions and all that, but you also want to be very good at being organized, being a good communicator, being able to be a good team player, because nobody can do everything by themselves. And one of the other big discrepancies between, I think, academia and industry is documentation and paperwork. There's lab notebooks and things like that you tend to take care of when you're doing research experiments. But when you're in a company that has certifications and standards that you have to stick to, even just making a cross-out can all of a sudden be a problem on an audit. So I think just those are some ideas that I have for, ways to kind of give you guys input into how to, teach these students from that workforce perspective on what might differ once they get into industry. So as I'd mentioned, we had the general kind of internships in the past. So now that we're under Antilia, we just have our very first student, and we're actually calling it a fellowship. So instead of spending, say, their whole summer in virology or in bacteriology and learning more just about those technical skills, this time the student is actually going to spend about 2 weeks in each department in the company. So they're going to get a full-blown overview of all the different departments. So it'll be, the science departments, the regulatory department, the legal department, the customer service department, which is really going to help them see, from a 50,000-foot level, all the different things that go into taking an order, making a product, quality, documentation, things like that. So I think as, zeptometrics relationship under Antilia matures and our ability to interact with you guys, I would hope that in the future we'll be able to, set up something, whether it's in person. Obviously, COVID's had some issues on, people being in person for certain things. But I would love to expand the relationship with your institution as we go along. The other thing that I wanted to mention, not to digress too much, is the other part of workforce development is sometimes students come in and they have their idea of, being, say, a pharmacist or a doctor, or basically one of the common career paths that are out there. But there's really so many other career paths that just aren't broadcast because, you can't solve a medical case in an hour on TV like they see on the TV shows or, the cop shows and things like that. That's not reality. There's so many other people behind the scenes that are running tests or developing tests, growing viruses like we do, things like that all kind of build into the bigger picture of certain career paths. So I just think that also with the workforce development and conversations with industry and things like that, to try to find out for these students, what other possibilities exist other than those, run-of-the-mill doctor, lawyer, kind of pharmacist, career paths. There's so many other things, and if they get the chance to be hands-on, not every scientist loves doing science. Some people would rather be at a computer doing data analysis. I've had conversations with kids who love math and they love science. It's like, well, think about biostats or something along those lines, because with genomic sequencing and all that, I think the pathways that have existed are changing, but there's also new career paths that are emerging as technology continues to grow. So, thank you.
That's wonderful, and I really appreciate your answer because it really, it's very important because that's always I talk about workforce development and the importance of workforce. If you look, and I don't know if you agree with me, our graduates, majority of them end up taking jobs at the biomanufacturing industry because the industry is growing and the technology is, changing by day and by month, and so there's nothing more important than preparing students to be having these soft skills that you mentioned. That is not part of the curriculum under an undergraduate major, whether it's engineering, life sciences, pharmacy sciences, whatever. Those are the soft skills that you mentioned that the industry wants, and I think that may be the new way of redefining curriculum by academic institutions to add few of those. I am one who added, a lot of, I teach cell culture for many years, and I added all those elements of cell culture that's needed for, biomanufacturing industry, scale up, different methodology for scale up, GMP. And so I think this will be something, but I am really excited, Dr. Delisa, about your technology. Now, I am really excited, that this is cell-free biomanufacturing. And then it's coming out of a prestigious institution with a distinguished professor that we have here. It is really an area where really when we talk about workforce development and when we talk about, bringing the workforce, this technology is coming. So from your perspective, and you are director of the Center for Biotechnology at Cornell, how do you see us getting into preparing some of these, students? Perhaps there will be a spin-off. We don't know. You may be a spin-off next year. We don't know. And spin-offs have been the backbone of the biomanufacturing industry from institutions like yours and MIT's and Stanford or what have you. This is the area where it's highly specialized area. But we don't have any of that in our curriculum. We don't have any of these soft skills, even in the training programs that are ongoing now. And so the question for you, Dr. Delisa, what is your solution for that? Is the center, your institute is going to be involved in some workforce training so that you can provide the labor force for these companies that pop up from this technology?
Yeah, so great question. I think, first let me say, at the core of your question is, especially in life sciences and in biotechnology, how do we give students the skills, training, and the to be able to make an impact. And cell-free is just one example. There are many, many technologies being developed in academia, in industry, that are new and will require new ways of thinking, new approaches, new infrastructure potentially. And so I think as those things are happening, we have to think about, what are the baseline skills that students need that may not even be directly related to, let's say, cell-free technology, but are transferable across technologies. Another thing we have to do is perhaps look at how training happens in other disciplines. And not the formal training in the classroom, but the perhaps less formal training that's equally, or some might even argue more impactful. So I like to think about, if you look at computer science on our campus, or mechanical engineering, this notion of maker spaces is one that pervades some of these fields and gives students at very early career stages the opportunity to put their hands on things and tinker and learn, and as we heard earlier, make mistakes, right, where all the real learning happens. It's really difficult to do that in life sciences, at least on an academic campus, right? We don't have too many examples of biological maker spaces, but we need more. And I think actually the center that you built here is along those lines. It's thinking in ways, for how do we put students in an environment where they can put their hands on technology, on capabilities. The focus here may be, CHO cell-based manufacturing today, but it could be cell-free technologies tomorrow. But the infrastructure is really what's critical. Having access to these kinds of spaces where you can flexibly reconfigure them as technology changes and as interests of students change and as industries' needs begin to change. So I think we really have to, universities are constantly looking for ways to create more hands-on experiences. But as has also been brought up here this afternoon, that takes money, it's expensive, and I'm not, I think it's unfair for academia to cry poor, but certainly, creating these spaces from scratch is a pretty significant institutional commitment. And it's not just building the infrastructure, as we can see here for CBET in the center, but how do you maintain that, right? It's often feasible to get the investment initially to build something, to put equipment in place, but how do you maintain that equipment? How do you buy the materials and supplies? How do you pay for all of that? And then how do you keep it, how do you renew it? Because as we've already been talking about in the vaccine or the cell-free technologies, technology is moving so quickly. And it's moving perhaps not as quickly in biomanufacturing, but it's still moving. And so it's a moving target, and it's difficult when you invest heavily in something, the fear that what you just bought is going to be obsolete in 5 years, in 3 years. That's a concern that all universities are struggling with in terms of creating core facilities, whether it's for training or even for providing services. I direct the Biotech Institute at Cornell, which handles genomics and proteomics and bioinformatics and all of these great capabilities that support academic research. But it's a nightmare thinking about how you do this sustainably, and if a piece of equipment goes down or you buy it today and it's obsolete next year. These are I think real challenges, and I think solving these is largely an academic problem, but I think as we're hearing today, there's room for academic industrial partnerships to perhaps lighten the load, to create what's needed perhaps for industry, and maybe industry helps support that process, but backed by academia in a way that's preparing students to deal with these really exciting and game-changing new technologies. So I don't know if I answered your question exactly, but I think it's about...
And it's understandable, and I think I'll come back to Johan. One thing that comes to my mind, well, I've been doing training programs for over 35 years, So really, you don't see any really recognized company out there that are doing what we are doing it in academic institutions. Do you think the reason for that, and you can chip in into this, or on your view, Ali or others, you can chip into that, academic institutions are institutions that are really teaching. We do teaching. And so why don't leave teaching for us to do, right? And so the question that comes now is say, okay, so we do teaching, we excel at teaching, and so we can teach those soft skills. So now how about, and then Ronnie can pitch into this one also. I mean, there's something I called it door to floor some times ago. It's becoming a common thing now. It's door to floor is that when you get a job at the biomanufacturing industry, you enter the door, you're not allowed to touch anything on the floor until you get through all those trainings on SOPs and what have you. And that can take months in some industries. It could be a month, it could be 3 months. So John, what do you think with all your contacts at Nimble and all these industries that are part of Nimble, is it possible that these companies can outsource part of these training programs that they do in an intense week, for example, at BTEC or at CBET or somewhere else that they choose, and then we can really reduce that door-to-floor time. Instead of being a 3 months, it could be a month. That saves them 2 months of production. That's millions and millions of dollars. Can you work with your industrial partners to think about outsourcing? Because then we're talking about outsourcing production into outsourced companies like CDMOs. But how about outsourcing some of those training programs to these academic institutions that put a lot of effort, and they are teaching institutions? So, we know how to teach, really. So we can do the best, and you tell us how you want it, and we do it.
So I want to comment. I can answer your question, but then I also want to go back and piggyback off the last two comments.
So, yeah, it's a discussion.
because it's relevant. And so the short answer is yes. I mean, and at BTEC and for that matter, at BETC, at the facility down in Texas, at NCTM and down in Texas. I mean, we all do a lot of training for industry. We all do a lot of customized training for industry. And so that's happening. Companies are coming to us and saying, hey, we need training in this, we need help in this. A lot of it is not, and I think that's where some of the different centers are somewhat specialized in how they approach it. Like at BTEC, we don't do operator training. We don't sit there and run, we have, but our sweet spot is not running cohorts and cohorts of operators through to give them truly the operational knowledge to do it. We do a lot of what I'd call applied theory, and so it's giving people, sort of that foundational understanding scientifically of what's going on in all the different unit operations. And so it's a lot of, scientists, engineers, advanced roles in manufacturing that we're training. And so we do, and I say we collectively with all the different training centers that are out there. Everybody's doing this. Everybody's doing a lot of training for the industry. I think there's a limit to how much of that training can be outsourced versus the companies what they need to do in-house. And I think I used to ask that same question of how can we do more of this here, but the question I keep asking myself more recently is like, how can we work in a paradigm where a lot of the training needs to happen on site? But I think, and that's why I want to come back to the other questions, because the other thing that academia does in addition to teach is it inspires. You're there to inspire students. And going back to Kelly's comments and about getting students with a lot of different disciplines aware of the industry, that's critical right now. And I think that's like a soapbox I get on all the time. Any of the students that are in this room or any of the students that are at BTEC or any of these facilities, they've got it made. They're already aware of the industry. They're going to go jump right into process development, material science, or manufacturing science or one of these other areas. They're not going to go work as an operator. The industry still needs all those operators. The industry still needs data scientists, computer scientists, mechanical engineers. They need all these other disciplines that right now, so many of those students, pharma isn't on their radar screen. And so I think there's a role for all these centers and to do a better job at, how can you, all those students don't need semester after semester or course after course of hands-on experience. And so from a cost perspective, it's not expensive. They need a brief little glimpse to say, hey, this industry is exciting. And that's not expensive to do, but these centers are well set up to do it. And I think that's a critical piece, and just sort of, there's a huge mismatch, or there will be a huge mismatch between supply and demand over the next 5 or 10 years.
Thank you. Ron, you want to say something?
I think it's true. There is a huge mismatch, and I think there's a lot of there's a lot of debate at every company on where there's the opportune place to train. And, you know, there's a lot of quality organizations at many companies that would, you know, refute getting the training from an external source. So it is a battle, especially when you start talking about people working in GMP facilities, turning valves, making decisions that are relevant to quality, right? So can they be outsourced? I don't see why they can't be. And I'll go back to what we were talking about with the next wave of industry when we get more digital. I've seen training in virtual reality, right? Where people don't even step foot on the floor in their training. So why does that need to be done at a site? Why can't that be done at a learning place? So I think the growth is there. I think the market is there. I personally, you know, I also think let me back up before I say this, but I really think that the gap that we have too is on the requirements around education. There's different education levels for different jobs, but they're not exclusive. Entry level is really the only thing that you need an education to get into, and that's an artificial thing too. We don't need to get into that. But the training courses for operators, they should be different, because you don't need an operator with a 4-year degree. You need someone hands-on. Much like you don't need a mechanic that's got a bachelor's in philosophy. It doesn't really matter. You want him to be mechanically sound. That's what a lot of these operators need to be. And I know somebody mentioned NIBRT, and there's many facilities in Europe that train people to be operators, and maybe they're more educated, but it doesn't matter. In Bristol, many years ago, we actually partnered with Onondaga Community College to run I think it was a 16-week or an 18-week training course for part of their program. So it was an actual course for associate level scientists and engineers. Worked fantastic. We got 12 people out of that program. And that's what we needed at the time. It's really just a matter of, fit for purpose. I believe personally, we waste a lot of time making people train on things they don't need to train on as well. And I mean, that's where I get into my personal opinion, where if I send someone to a course, it's got a curriculum, they train on it. Much like I send my daughter, next year I'll send her to college, right? She's a senior. I don't go in and audit the college to make sure, right? I mean, I'm not going to go talk to Matt and say, let me see your syllabus before my daughter signs up for your class. I think that's some of the things we need to think about. Is we need people who come out of these colleges, come out of these training courses, to be able to not remember everything they did, but to remember why they did it and remember the general thoughts and the rationale, right? I mean, we all know scientific principles in math, but when you're doing a batch record and your balance gives you 5 decimal places, even though you know 0.04321 is not an accurate gram, right? And a scale that's measuring in grams and kilograms. But if you don't teach the person, they're going to write down everything. And then, you're going to be left with, well, that third digit was wrong. Transcribed error or something. So you have to learn how to learn at college at these training courses, and you need to learn broad strokes, and then you learn the rest on the job. And there's no quality person going to sign off on your learning of that, because that's what we call experience. And that gets back to what I was talking about earlier with internships and co-ops and things for if there's any students left. This is your opportunity when you're in college. Learn as much as you can where it's technically free. You can make mistakes and people help you, because that's where you're going to get everything. And that's one of the reasons why we send people to these courses, because they can learn in a safe environment.
Great.
Anyways, soapbox.
Anyone wants to come in? Any of you on board, anyone wants to chip in to discuss that? Derek, you have something to say? Okay.
No, I think that's perfect.
I think I asked all the questions I had in my mind. But if you have a question in the audience or online, I don't know, Julian, Ihsan is there? Oh, somebody have? Okay.
I was just going to chime in a little bit and say I 100% agree. it would be, my creative brain right now is in overload trying to think of some way to streamline what you're asking for. But I think it's complicated because, even if you're reading SOPs or you're doing, say aseptic technique with cell culture or making reagents or things like that, if you read about it, that's one thing. But science is really a hands-on learning experience. So I can have a new employee or an intern come in and read for 3 days in a row, and they're not going to retain anything until they are partnering that SOP or that batch record with what they're really technically performing, right? That's when you're really going to learn. I like to say you can't learn science from a book, right? You need a good mentor. You need somebody to pass that torch so that you can hands-on really learn those technical skills. So, some of my thoughts would be, even remembering through college and grad school, things like that, you're going to learn certain techniques. You're going to become a master in some cases of certain techniques. It doesn't mean that you're really going to land a job always using those techniques. So if you have a way to maybe partner somebody's, somebody's ready to go to, a certain field, or maybe they've already co-opted and they're lined up for a certain career, that really helps them and you and, the employer in the end, if you can focus in on those specific techniques and skills that are going to be required. But I think, really knowing exactly what you're going to do is difficult. So how do you pre-train somebody? It's an open question. It's something that I'm definitely going to spend some time thinking about. But again, like when you have things you have to sign off on, and like for us, we're working with bio-level 3 viruses. It's not even that I can have you teach somebody that outside of our particular lab setting. So there's definitely some barriers to doing that. But I also think that there's some way to maybe address some of those certain training techniques, safety trainings and things like that, that might be more general to specific fields. So just my 2 cents.
That's a great point. Thank you very much. Now, Dr. Madini, is there any question from the audience or from the online?
Absolutely. Thank you, everybody. Any questions from the audience? The mic is open. And if you will do one, or I can bring the mic to you if you don't mind touching it.
Any from online?
No follow-up from
No one from online. Well, let's see.
Dr. Rashid, if I may, can I make a couple more points while we are waiting for questions?
Yeah, go ahead.
Well, I enjoyed this discussion. I think most of the things we all agree, that's great. Right, and there is a great consensus that hands-on training is very important to bring fundamentals in action. Of course, industry do their parts to a certain extent, bring the students for internship or fellowship, all this co-ops, that's great. But I think industry has a responsibility to help academic institutions to equip our labs, our facilities, our centers, because universities don't have that much money to invest. They can invest in building, but when it comes to the equipment, they run out of the funds very quickly. So I think we cannot expect only the equipment manufacturers to help us to equip our labs. So I'm just going to bring these great examples that we have had in the recent years. This company, as many of you know, CSL Behring, happened to be in our state. So they had a good relationship with our university. They invested $5 million at our university. So we use a significant portion of this money to renew our fermentation pilot plant. Of course, the equipment manufacturer give us a good discount. They did their part. They even donated a couple of the equipment on top of it. But with their funding, now we have a state-of-art facility that we can train our students. When they go to the industry, they are going to be using similar equipment. So it is a very good example that all pharmaceutical or biotech companies, if they really want a good educated workforce, they need to put their hands on under the stone a little bit and help the academic institutions to equip their facility. So internships are great, of course, but there are another opportunity that university and industry work together, which is not used very efficiently yet, the capstone projects. So every major, or most of the majors in their senior year, every student has to go through this capstone experience. Of course, as the instructors, we create hypothetical problems, hypothetical projects for them to work on it. Isn't that excellent idea to industry bring those problems to the table so we can have the team of students work around that problem while they are in the school using the facilities that we already established. And this is a one-year project, basically. It's much, I think, low cost than the internship or faculty sabbaticals or etc. So students don't need to move anywhere with this technology that we have today, like Zoom and all other things. They have weekly meetings and reporting and etc. So I think we need to look more closely at these capstone project ideas. Then it will prepare the students' industry problems ahead of time, applying and solving those problems hands-on, most of them. And industry will have a chance to get to know our students better and choose the ones that they like for the full-time job. So I think capstone projects that we offer, industry should take advantage of that for their benefit and helping us to make that relationship another level. That's what I would like to share.
Well said, Ali, and actually we have capstone projects going on right now at the center. Your student, Dr. Mahdina, having a couple of students that capstone, I have a couple of students. So we're doing capstone. Dr. Manakshi has been doing capstone for a good number of years. It's really the way we go, and the center is a very helpful factor in doing these capstone projects. And we already have a student who did a couple of internships at Eli, and then she got an offer before graduation. So these things work,
That's great here. So we need to do more of that at various institutions.
I think if there is any question from the audience or any comment from our distinguished speakers, we're just about 4 o'clock. We can go 5 minutes more. Okay. If there are questions or if you want to say anything before we wrap it up, anyone who has any point, please. Derek, you have something to say?
I can. I think, when we think about biomanufacturing, if you look at the last 5 to 10 years, we've had more therapeutic modalities open up than I think in a number of decades beforehand. If you think about it, we've moved from small molecules to antibodies to proteins and now wholesale therapies, gene therapies, and the pace of innovation for the latter two categories is just blistering. The amount of capital that's been deployed into cell and gene therapy companies is amazing, both public and private capital. And these are all brand new. There's no decades of experience in cell and gene manufacturing because they haven't been around that long. So I think this really opens up an opportunity for innovation and best practices and a lot of places for collaboration to really move those fields forward. So, I think we're in a pretty amazing time in the industry where, we have, there's a lot of capital out there, both in the private and public markets. You're seeing a lot of new companies formed and a lot of things formed directly out of academia. And it's made for, I think, a really fertile environment for collaboration. So from my perspective, I think there's probably a lot of opportunities to think about what really the next wave looks like in terms of biomanufacturing. What are the things that industry is going to need? What are the niches that academic advancements can fill, and that, in particular, collaborations with institutions like yours can help move the field forward?
Thank you so much. That's what Dron said about customized programs. We have customized programs. We already have, because this is a trend now, custom design for company X, based on their management's desire when they have a shutdown, for example. So we do that. I think all institutions that are involved in education and training doing customized, which is custom design, is really good. But I think, Julian, you allowed another minute, or we're done? You're the boss, what do you think? Wrap it up? Yeah. Okay. Well, thank you so much for all our panelists. Let's have a hand for our panelists.
Thank you.
We really appreciate your time. We appreciate you traveling, coming all the way here to help us out with this panel. Hopefully, This was a good interaction discussion that we did today, and we learned a lot from this. And as we plan for future of CBET, there are a lot of good points that have been brought up in this discussion that can become part of planning of CBET in coming years. Yeah. So thank you very much. Thank you all on distance. We appreciate your time. We appreciate your efforts, and may we see you again in some of our programs.
Yeah. So.
Have a great evening. Thank you.
Thank you.
Bye-bye.